All right, let's dive into it. Class, in this recording, we're going to focus on some membrane-bound organelles. First on the docket, we have the lysosome. And then when we think of the lysosome is a package of enzymes. And these enzymes are going to be bound up together by some phospholipids, some membrane. Generally speaking, the lysosomes are round, but it is possible that they're a little variable in shape. Our lysosomes you can think of as the recycling plant of the cell or the digestive center of the cell. They contain a lot of hydrolytic enzymes. And if you recall from earlier in our chapter 2 discussion when we reviewed chemistry, hydrolysis is when we use water to break apart the covalent bonds that hold biomolecules together. So we can have hydrolysis of proteins, nucleic acids, complex carbs. We can take these big macromolecules and chop them up into little pieces using a lysosome. And while we're using that lysosome, to chop up molecules, there are different forms of chopping up or different reasons to chop up molecules. We can have autophagy, so maybe there's an organelle that's non-functional, and that non-functional organelle needs to be broken up into little pieces and recycled. It's kind of like taking your old Lego sets, ripping them up into little Lego bricks, and then recycling those Lego bricks to make a new spaceship, or to rip apart your Lego spaceship and make a castle out of your Legos. We will reuse those subunits, those monomers from those macromolecules. We also could have autolysis. Autolysis is referred to as cell suicide. Every once in a while, a cell just needs to self-destruct. It may be a precancerous cell where for the overall good of us as an organism, we need to have that particular cell kill itself. And it will destroy itself so that we don't have a cancerous tumor form in our body. Or maybe the cell has been hijacked by some virons or viruses and is now pumping out lots of viron particles. And in order to mitigate the amount of damage that occurs to us as an organism, that virus-infected cell will destroy itself through autolysis. Now, as we're looking at our next organelle, the peroxisome, peroxisomes and lysosomes physically look very similar to each other. Usually the only way to tell them apart on a model in lab is just to look at the color. The key difference between them are the enzymes produced by these, also where they are produced. Peroxisomes are going to be produced at the endoplasmic reticulum. And as we look at these, the light peroxisome is going to use molecular oxygen to oxidize molecules. So when we think of peroxisomes, I want you to think of oxidizing it. Peroxisomes have peroxides. They have lots of hydrogen peroxide. They also are going to have superoxides as well. Within a peroxisome, we are going to have an enzyme known as catalase, one of my favorite enzymes out there. Catalase does this wonderful thing when you mix it with hydrogen peroxide. It bubbles. And if you mix it with hydrogen peroxide and dish soap, you can have a stream of foam shooting out of your beaker if you want. It's a lot of fun. I remember doing that on a city bus in Duluth once. Whew, that was a mess. I digress. So as we're looking at the peroxide is a free radical that's present. It's just a natural byproduct of making ATP. And it's bad for us. We don't want hydrogen peroxide floating around our cell. So the catalase within the peroxisome will neutralize the hydrogen peroxide, or the free radicals that we have floating around in our cells. Peroxisomes also help us with detoxifying alcohol and other drugs and oxidizing toxins. And when we say oxidizing, usually that means attaching a oxygen molecule covalently to that molecule, or that or an oxygen atom covalently to that molecule that we're oxidizing. Or in a more technical sense, it can mean that we're pulling electrons away from the molecule. We also use peroxisomes to break down fatty acids into individual acetyl groups, and those individual acetyl groups are then going to be sent to the mitochondria to make ATP. That's referred to as beta oxidation. Our peroxisomes are present in all cells, but they're especially abundant in liver and kidney cells. Oh, there's so much I could say about that, but I'm not. For the sake of time, we're going to move on and look at our electron micrograph of our membrane-bound organelles. So here we can see some lysosomes present. Those lysosomes are near some mitochondria. And then here we have some peroxisomes present. Note how the lysosomes and the peroxisomes are very similar looking in appearance. It's difficult to tell them apart, and I'm covering up the sense of scale here. So here, this is 0.3 microns versus 1 micron, so we're much more zoomed in on the right compared to the left. We also have proteasomes in our bodies. Proteasomes are a hollow, non-membranous organelle that is going to dispose of surplus proteins. So here's our protein that's being designated for destruction. We're then going to tag that protein, and once the protein is tagged, it's then going to be assimilated, processed by the proteasome. And that proteasome will take the protein and remove its tertiary and secondary structures and get it down to just the primary structure, the spaghetti noodle or beads on a string of amino acids, and pull it in and feed it through, and then snip, 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 chop it up into little peptides and or individual amino acids so that we can reuse and recycle those amino acids. We also have a mitochondria in our cell. The mitochondria is a plural. This is a plural form of the word. Mitochondrion is the singular, and I know I and many other highly educated biology professionals mix that up frequently because typically people who are really good at biology aren't very good at grammar. It's hard to find people that have both skills. So as we think of the mitochondria, it's the powerhouse of the cell. It's used for making ATP or synthesizing ATP. It's primary function. We get our mitochondria from our moms, our mothers. We have maternal inheritance of mitochondria. So there's some really interesting things about mitochondria. For instance, mitochondria have their own genome. Mitochondria have their own ribosomes, their own separate protein synthesis. The DNA within a mitochondria is called mtDNA, mitochondrial DNA, and it's used by it's going to be sent to the mitochondrial ribosomes to make mitochondrial exclusive proteins. As we look at the mitochondria, it has a double membrane enfolding it. The outer membrane is typically going to have an elongated or kidney bean shape. The inner membrane is going to fold back and forth with some cristae. And in between those cristae, the innermost part of the mitochondria is a region called the matrix. And within that matrix, that's where we have all of those interesting things happening. As we look at the mitochondria, it functions to take organic energy, particularly energy that's stored in a carbon-hydrogen bond, and repackaging that energy to be put into a phosphodiester bond within an ATP molecule. And then that ATP molecule is going to be able to carry that energy from the mitochondria to power a chemical reaction, whatever our favorite chemical reaction may be at that time. So here we have a fig from your textbook showing a mitochondria. Here's an electron micrograph. We have a rather complicated organelle right here. It's believed that mitochondria at one point in time used to be a completely separate organism that formed a symbiotic relationship with the cells that make up us and all the other eukaryotes out there. We also have centrioles within our cells. And when we think of a centriole is going to refer to one short cylinder of microtubules. And there's going to be 9 groups of 3 microtubules within each centriole. If you take 2 centrioles, make them perpendicular to each other, and put them next to each other, then you have a centrosome. And the centrosome is an area within the cell that is going to be producing the cytoskeleton and play a very important role for cell division. In addition to forming the basal bodies of cilia and flagella, the centriole is going to be the origination point of the microtubules and the mitotic spindles that help organize the organelles and the chromosomes within the cell. Here we can see our groups of 3 making up a centriole, and then 2 centrioles combined makes up a centrosome. We also are going to have inclusions within our cell. To review, an inclusion is going to be storing a foreign substance within the cell. So we can have glycogen granules. Oops, let me go back here. There we go. So we can have glycogen granules. Glycogen is the animal complex carbohydrate. We can store pigments. We can store fat. We can also store viruses, intracellular bacteria, debris that's been phagocytized. So there are different kinds of inclusions. We can store different kinds of things within our cells. We should emphasize here that it's never enclosed in a unit membrane, and it's not essential for survival. So inclusions aren't always going to be present within the cell. Pragmatically speaking, it's very difficult to tell inclusions apart from phagosomes and lysosomes. As recently as the 2000 teens, early 2000 teens, I want to say it was 2013, there was a new organelle discovered. And it was one of those membrane-bound sacs that was difficult to distinguish between the inclusions, the lysosomes, and the peroxisomes. So we're still learning more about the cells as time progresses. This is, if you want to watch an excellent review video, you should go onto YouTube and watch The Inner Life of a Cell. The full version is about 8 minutes long. It initially came out in 2006. And to me, it's the gold standard of movies. Not only does it have excellent background music, but it also does a great job of showing how the different organelles interact with each other. I would play the recording in this this recording I'm making right now, but it's copyrighted information, so I can't include it in this particular recording. You have to go onto YouTube and watch it yourself. That's all we have for our recordings on Chapter 3 on the cell. If you have any questions or comments, please feel free to post them on the class discussion board or shoot me an email. And as always, happy studies.